Alright, hello everybody. My name is Ashley Fowler and I'm going to be presenting to you my paper entitled Phosphorus Content of Cool Season Grasses as Affected by Stage of Maturity and Fiber Fraction. Phosphorus is a very important mineral for both plant and animal life. However, recent environmental concerns have been raised regarding phosphorus usage. Phosphorus found in fertilizers and animal feeds is mined from rock phosphate reserves. And in 2007, Derry and Anderson modeled world rock phosphate production from the early 1900s to a value in 2050 that they predicted. And they predicted that peak phosphorus production occurred during the late 1900s and is expected to dwindle to very little within the next 50 to 100 years. On the other side of the phosphorus cycle, excessive phosphorus excretion in fields and pastures in the form of fertilizer or manure can increase the risk for phosphorus runoff into nearby water bodies, causing eutrophication as seen in this picture, and also water pollution. So in an effort to reduce phosphorus supplementation as well as excessive phosphorus excretion, we were interested in determining if pasture plants could provide enough phosphorus to meet animal requirements. In a grazing system, pasture grasses can provide most of the nutrients required by grazing animals. So it's important for producers to understand how much and also how available those nutrients are in the pasture so they can make sure their animals are getting the nutrition that's required. In horses, we estimate phosphorus digestibility to range from around 35 to 45%, but these values were measured in horses fed diets of hay and grain, and little is known about the phosphorus availability in pasture grasses. As pasture plants mature, the quantity of cell wall increases, and this increase dilutes out other important nutrients such as phosphorus, which leads to this decrease in quality that you typically see. NDF, a component of the cell wall, is negatively correlated with digestibility in animals. So it's possible that phosphorus that's associated with this less digestible fiber fraction could also in turn be less available for digestion. So stage of maturity could potentially influence not only the amount of phosphorus, but also the availability of that phosphorus. Which leads us to the objectives of the current study, which are to characterize changes of phosphorus content of cool season grasses throughout different stages of growth, and then also to determine if the phosphorus content in the fiber fraction of these grasses changes throughout the different stages of growth as well. And lastly, we also were interested in looking at the in vitro phosphorus digestibility of these grasses and if it's related to NDF-associated phosphorus. For this study, we used two cultivars of both the tall fescue and an orchard grass, so Cajun and Bronson for tall fescue and Persistent Prophet for orchard grass. These grasses were seeded in September of 2013 using a disc drill and were fertilized with nitrogen the following spring. Our plots were 5 by 15 feet and were arranged in a randomized complete block design with 3 replications per cultivar. We took samples at 4 time points within the growing season. We took them at early maturity and at maturity. The plots were then harvested, and we took 2 samples after harvest to measure regrowth. We took representative samples randomly throughout the plot and clipped them at 2 inches from the ground. Once we had our whole plant sample, we then subjected them to sequential analysis for NDF and ADF using the ANCOM fiber analyzer system. And then we took samples of our whole plant, of our NDF residue and our ADF residue, digested them using the Kjeldahl method, and then analyzed each of those fractions for total phosphorus. We also measured in vitro phosphorus using methods that simulated equine foregut or enzymatic digestion that occurs in the stomach and the small intestine, and also foregut plus hindgut digestion, so it also includes that fermentation that's going on in the latter part of the equine digestive tract. We used duplicate samples of each rep per cultivar at maturity. For the enzymatic digestion, we incubated the feeds in a pepsin and then an amylase solution. And for the fermentation, we use the ANCOM Daisy 2 incubator with equine feces as our source of microbial inoculum. We then measured phosphorus in our samples after the digestion to estimate in vitro phosphorus digestibility. Data were analyzed using a repeated measures ANOVA with rep, cultivar, and sampling day as the main effects, and means were separated using an LSD test. We made our comparisons between the enzymatic and two-step methods using a paired t-test. Significance was set at p less than 0.05, and a trend was considered when p was less than 0.1. Moving on to our results, this graph is showing you whole plant phosphorus across the sampling days. So sampling days on the x-axis, with E being early maturity, M is at maturity, and then regrowth 1 and regrowth 2. So we can see that whole plant phosphorus starts off high at early maturity, decreases when the plants arrive at maturity, and then rebound during the regrowth. When we look at whole plant phosphorus among cultivars, we see that tall fescue Cajun is significantly lower compared to the tall fescue Bronson cultivar, as well as the orchard grass cultivars. So this graph is showing you whole plant phosphorus among cultivars along the x-axis, and then also at each of the different stages of maturity. And we did see a significant interaction between sampling day and cultivar, which means that growth stage affected each cultivar a little bit differently. So for example, you can see that the tall fescue Cajun, shown here, did not change across the sampling days. But if you look at the orchard grass, persistent profit, those have much more fluctuations with regard to the growth stage. Looking at NDF across the days, we saw what we expected at early maturity. The NDF was low compared to when the plant was reaching maturity, and then it stayed high throughout regrowth. Among cultivars, we saw that the tall fescue cultivars were both lower in NDF compared to their orchard grass cultivars. So to remind you, once we had our NDF residue, we then analyzed it for phosphorus to look at phosphorus located within the NDF fraction. And that is shown in this graph. So we see that the phosphorus in the NDF fraction starts off high at early maturity, decreases through maturity, and stays low throughout the regrowth stages. So kind of the opposite of what we saw with our NDF percentage. Among cultivars, however, we see again that the tall fescue varieties are lower compared to the orchard grass varieties. Now this graph is showing you the percentage of the whole plant phosphorus that's located within the NDF fraction. So at early maturity, NDF phosphorus represents about 5% of the total plant phosphorus. It increases slightly at maturity and then decreases to around 4% of whole plant phosphorus throughout the regrowth. Among cultivars, we see again that the tall fescue hovers around 3 to 3.5% of the total phosphorus in the NDF fraction, and the orchard grass varieties are significantly higher, around 6 to 6.5%. So even though we did see significant differences between cultivars and also among sampling days, it's a relatively low amount, less than 7% in all cases of the total phosphorus that's present in the NDF fraction. So that means that most of our phosphorus is located in the cell contents. And as I mentioned before, we also measured phosphorus in the ADF fraction, but it was not detectable, so I don't have a graph to show you for that one. So for discussion, we saw that whole plant phosphorus was high at early maturity, decreased at maturity, and then rebounded during the regrowth phase. And a possible explanation for why we see this rebounding is that phosphorus can be mobilized from the crowns and roots of plants after defoliation to help support the new tissue growth. We also saw that phosphorus associated with the NDF fraction decreased throughout the growing season. However, because most of the phosphorus measured was located within the cell contents, we expected that phosphorus availability would be high for a grazing animal. So if we looked at our total tract phosphorus digestibility using equine feces, we didn't see a difference among cultivars for phosphorus digestibility, but phosphorus digestibility was relatively high, ranging from 87.5% to around 91% digestibility across the total tract. However, horses are unique in that they absorb phosphorus in their hindgut as opposed to in the small intestine like most other species. So we wanted to examine how much phosphorus would be liberated in the small intestine and the stomach so that the horse would have a better opportunity to absorb it. So we also looked at enzymatic digestion, shown in the yellow bars, compared to the total tract digestion, shown in the blue bars. And again, we didn't see an effect of cultivar on enzymatic digestion, but we did see that for some of the cultivars, namely tall fescue Cajun and orchard grass persist, there was a difference between enzymatic digestion and total tract digestion. And this means that the phosphorus that was liberated during the fermentation step was likely associated with fiber that the microbes were breaking down during the fermentation process. So as I mentioned before, horses consuming hay and gray diets have a phosphorus digestibility of 35 to 45%. However, we saw that using pasture grasses, foregut digestibility was greater than 75%, which is a lot higher than 35% used by the NRC. And across the total tract, we see it's greater than 85%. So phosphorus from pasture is highly available for digestion. So what does this mean for our grazing animals? This graph is showing you whole plant phosphorus, shown in blue bars, and then NDF phosphorus, shown in the yellow bars, across the grazing, the growing period. So if we have an animal at maintenance who requires around 0.15% phosphorus in his diet, his needs are going to be met throughout the growing season for all the pasture grasses that we sampled. And if we have a lactating animal, even in most cases, their needs will likely be met if the grasses are kept in a vegetative regrowth stage. Those animals will most likely be okay as well. In conclusion, we saw that most of the phosphorus in pasture grasses is located in the cell contents, which leads it to be highly available. We also saw that phosphorus content is affected by cultivar and stage, but it's usually sufficient to meet the needs of animals without additional phosphorus supplementation. So a more sustainable approach to grazing might be to develop a strategic supplementation program that's designed to complement the phosphorus that's already available in the pasture so that we can decrease excessive phosphorus utilization and excretion. I would like to thank a variety of people for helping me with sampling and also with analysis. And with that, I would like to thank you for your attention and take any questions you may have. Yes.
Why do you think there's such a big difference between pasture grass and hay and phosphorus?
So the values, what's that? Okay, the question was, why is there such a big difference between hay diets and pasture diets with relation to phosphorus digestibility? I'm not entirely sure. The NRC uses those values as an estimate of true digestibility, so that's how much the horses are actually absorbing and retaining. What we did was we measured how available the phosphorus would be, so we're not measuring absorption and excretion. So it's possible that all this phosphorus is available to the horse for digestion, but they only really need, 35% of what's available.
So on the last conclusion, you said something about like strategic supplementation to complement this. So like, what would an example of strategic supplementation, what would that be?
So if you are having a cool season pasture grass and you have that bimodal distribution, maybe you might need more mineral supplementation during the summer when those are kind of slumping, and less supplementation during periods of high growth. Or if you're allowing plants to reach maturity, then you might need to supplement more as opposed to keeping it in vegetative regrowth states.
We have time for one more question. So when would a teamster introduce supplementation to his team?
A teamster?
He's driving his horses, they're working.
Oh.
So when would he supplement them? Or she?
Um,
I'm thinking that might be what he's asking, I'm not sure. I'm thinking about not minerals, I'm thinking about something else.
you mean like for
More energy than
energy?
in their roughage.
Mm-hmm. Yeah, you could use other grains and stuff. That's going to increase your phosphorus content, but usually energy would be more important than worrying about minerals at that point.
Right, well, let's thank Ashley.
Hello, today I would like to talk to y'all about the research that I have been working on looking at residual effects of herbicides used in pastures on clover productivity and establishment. So I thought this was important because, as you all know, there are many negative effects of weeds in pastures. Some of these effects are that weeds can decrease the quality of your desirable forages because they are often non-palatable or toxic to livestock. They can also decrease the quantity of your forages because they are often very competitive with your desirable forages. There are currently no pasture herbicides on the market today that are labeled for use in pastures with clovers or other forage legumes, though white clover has shown some tolerance to 2,4-D in the past. So the objective of my study was to formulate a plan that would provide for pasture weed control, but still include clovers as a forage component in pastures. I replicated my study in 2 locations in northeast Louisiana, one location being at the Macon Ridge Research Station in Winsboro, Louisiana, which is where I work, and the other at St. Joseph, Louisiana, at the Northeast Research Station. I chose 4 clovers for my study. The 4 clovers that I chose are the most popular clovers in our area. These are white, red, crimson, and ball clover. Here you can see white and red clover, and these are popular in our area because they have a longer growing season than most other clovers. And here I have ball and crimson clover. Ball is a popular choice because it's a prolific reseeder, and crimson clover is an earlier producing clover. So my study, the experimental design of my study was a randomized complete block design, and it had 4 replications. As I previously stated, I used 4 clovers, ball, white, crimson, and red. And I used 2 herbicides, grazing P plus D, which is 2,4-D and picloram, and pasture guard, which is fluroxapyr and triclopyr. I applied these herbicides at 4 fractions of the full recommended rate, and these were at 0, 1/4, 3/8, and 1/2 of the full recommended rate. I did this to simulate herbicide degradation in the soil over time. The herbicides were applied using a tractor-mounted spray boom, and they were applied at time of planting in mid-October. The clovers were planted with a drill-seeded planter, and it was a column-type planter that you can see here. Data collection was taken in the form of plant counts at Emergence and then again in March. These plant counts were taken in the form of, I counted every plant within 3 random 1-foot sections of row in each plot. I also took visual ratings at Emergence, and then monthly for 5 months. The visual ratings were on a scale of 0 to 10, 0 being no plants present, and 10 being complete stand establishment. I took clover yield measurements in May using a small plot harvester with a bag attachment, and data was analyzed using PROC mixed of SAS. These graphs show the effect of herbicide on clover stand at Emergence and then again in March. The y-axes show plants per foot of row, and the x-axes show the clovers. The blue bars represent grazing P plus D, the red represents pasture guard, and the green represents none. You can see at the Emergence count, there was no significant difference among any of the clovers, so this means that they all did emerge, and at Emergence, there was no significant difference among any clovers when treated by either of the herbicides. But then in March, you can see, if you look at the red graphs, or at the red bars, that pasture guard had no significant impact on the clovers when compared to the non-treated, but grazing P plus D significantly reduced stand. This graph shows the herbicide effect of clovers just 3 months after emergence, and the y-axis is the visual ratings on a scale of 0 to 10, and the x-axis shows the clovers again. And I'm using the same color scheme as before here. So the grazing P plus D, you can see by looking at the blue bars, you can see that grazing P plus D significantly impacted clovers, or it significantly impacted the clover stand even just 3 months after emergence. And the pasture guard had no significant impact. This graph shows clover yield response to herbicide rate. The y-axis shows yield in dry matter pounds per acre, and the x-axis shows the fraction of the recommended rate that was used. You can see when comparing the clovers to the non-treated that the crimson, which is represented by the red bars, was not affected at the 2 lower rates, but it was affected at the highest rate. Then white clover was not affected at the lowest rate, but was affected at the 2 higher rates. And ball and red clover, which are represented by the blue and the green bars, were negatively affected at all rates, though ball tended to be more sensitive to the herbicides. This graph shows the effect of herbicides on clover productivity. Again, we have yield represented on the y-axis by dry matter of pounds per acre, and the x-axis represents the fraction of rate used. You can see by looking at the blue bars, which are the grazing P plus D, that grazing P plus D, was the trend in all the others, significantly reduced yield, whereas pasture guard had no effect. So the previous slide, the effects that you saw on the previous slide were mainly due to the grazing P plus D and not the pasture guard. So these pictures show red clover and ball clover 1 month after treatment with a half a rate of pasture guard. And the pictures on the left, with pasture guard and grazing P plus D. The pictures on the left are red and ball clover treated with pasture guard, and the pictures on the right are red and ball clovers treated with grazing P plus D. So you can see that those treated with pasture guard they looked pretty healthy, and the ones treated with grazing P plus D are showing signs of herbicide injury. My arrows there show some of the twisting and other effects. And these pictures are basically the same as the previous slide, but this is crimson and white clover. On the left, you have crimson clover treated with pasture guard, and on the right, crimson and white clover treated with pasture guard, and on the right, you have crimson and white clover treated with grazing P plus D. Again, this is 1 month after treatment, so all clovers did emerge, but those treated with the grazing P plus D are showing signs of herbicide injury. These pictures show crimson, red, white, and ball clover when treated with a half a rate of grazing P plus D, and this is 7 months, a fourth a rate of grazing P plus D, sorry. This is 7 months after treatment. So this is the very lowest rate of P plus D. And so even at the lowest rate of grazing P plus D, you can see that we had significant stand loss, though crimson seemed to tolerate it a little better than the others. Now these pictures show crimson, red, white, and ball clover at a half a rate of pasture guard. This is 7 months after treatment. So you can see that even at the highest rate of pasture guard, we had no significant stand loss or injury. So in conclusion, clover stands and productivity were significantly reduced by grazing P plus D, even at low levels. So grazing P plus D would not be recommended for use in pastures with clovers. However, pasture guard did not significantly reduce stand or impact clovers in any way, and it would be a viable option in pastures with clovers. Ball appears to be more sensitive to these herbicides, while crimson appears to be more tolerant. I would like to thank my committee members and the farm staff and student workers at the Macon Ridge Research Station and the Northeast Research Station. And with that, I thank you, and I will take any questions that you have.
What were the active ingredients in the second herbicide that didn't injure the clover?
And the question was, what are the ingredients in the pasture guard? It's fluroxapyr and triclopyr.
Do you have any idea of the half-life for those herbicides?
I think that for the pasture guard, it's about 10 days, and for the grazing P plus D, it's about 90 days. I'm not 100% sure, but I think that's what it was. Yeah, there you go.
30 for P plus D, would you say would be the average half-life you see in studies on P plus D virus? That's 30. And so when you get into pasture guard is residual in the soil for 30 days, but the thing is it's not actually taken up by plants. So even though it's there, it has a half-life of 30 days, once it's in the soil, it's not taken up, so it's not going to affect the plant. Well, not significantly, it affects the plant.
Oh, and I'd like to point out that that this strictly was a residual study. It was not, in no way do I recommend that you spray these herbicides on your clovers. I mean, this was strictly residual, and it was all done prior to emergence. So.
Any idea of how much weed control you could expect from a half rate with these herbicides?
The question was, is there any, do I have any idea of how much weed control to expect from these herbicides? Well, that wasn't really what we looked at in this study, so I can't say for sure, but yeah, I mean, we didn't look at, this wasn't a weed control study as much as it was a tolerance study, so.
So on the pasture guard, what was your actual half rate? I mean, how many pints of pasture guard was applied?
Well, the question was, what was the actual half rate? So it would have been 1 pint of the pasture guard, because it's recommended at 2 pints per acre.
So, and is this the HL formulation of it that y'all use? High-low formulation, or was it the old-fashioned?
The question was whether it was the HL formulation. It is the current recommended formulation, so I believe that if that's what is currently, yes.